Good morning, students. In the last session, we have completed three topics: Mendel's experiments and his observations. Second topic it was Mendel's laws of inheritance, and also we studied incomplete dominance. But in this class, I am going to explain different topics. One is codominance, second one is test cross, third one is pleiotropy, the next one is polygenic inheritance, the next topic chromosomal theory of inheritance, the next linkage and recombination, and the last topic is sex determination mechanism. So we can start from the first topic. First is codominance. What is meant by codominance? So yesterday we studied two terms, dominant and recessive. We told dominant means which express outside, that is dominant character. Recessive means it which is hidden. Here codominance means both are dominant, both are equally dominant or expressing outside. So what is codominant? The alleles or genes which are able to express themselves independently even when present together, they are called codominant alleles. So there is one pair of genes or alleles, they both express themselves independently. Okay, if they are in together also. In the previous class we studied there is a capital W, only one W gamete will form and that will express. Sometime along with that there is one small w, I told that is recessive. Here both will express. Okay, that is called codominant alleles. The phenomenon is called codominance. So for this one example we can see, one is ABO blood grouping in humans. ABO blood grouping, you know which are the different types of blood groups. You learn A, AB, A group, B group, O group and AB group. So ABO blood group, it is controlled by gene I. This gene gene has three alleles, IA, IB, IO or small I. Both it is same, IO or small I. So here what is happening? IA and IB, it produce RBC surface antigens, sugar polymer A and B. Whereas small I does not produce any antigen. Clear? So IA, IB both produce antigens, but the small letter I, or I told it is also called IO, it does not produce any antigen. Then next point is IA and IB are dominant over small I. Clear? So IA and IB both are dominant over small I, small letter I. Hence IA and IB are dominant alleles and small I is recessive allele. Clear, children? So IA, IB dominant alleles, small letter I is recessive alleles. Then when IA and IB are present together, both express equally. That is called, I told, codominance. IA, IB are present together, both express equally and produce the surface antigen A and B, hence show codominance. So IA, IB both express equally and produce surface antigen A and B, hence show codominance. Then humans are diploid. Since humans are diploid, each person possesses any two of the three I gene alleles. Clear? So there are three alleles, IA, IB, IO or small I. So each person possesses any two of three I gene alleles, resulting into 6 different genotypic combinations, 4 phenotypic expressions. Clear? So these are the genotypic combinations and these are phenotypic expressions. That yesterday I told you, phenotypic, it means outward expression. Genotypic means inside or internal expression or inside expression. So how it is happening, we'll see. The phenotypic expression already you know which are the 4 types, A, B, O and AB. But the genotype you see, you can see here different types. So that I will explain first. Allele from parent 1. Here allele from parent 2. This is the genotype, this is the blood group. Here you see IA, both together form IA, blood group is A. Second, IB, genotype IB, blood group is AB because both express equally. Before I told it's codominance. Then here IA small I, it will small letter I, so it will form IA small I, but the group will be A blood group because small letter I, it is does not produce any antigen. Clear? It is a recessive, does not produce any antigen, it is recessive. Here IB, here the genotype is IB, the blood group it is B. Same like next one is IB small letter I. Here also you can see B is dominant, small I it is recessive. So the blood group it will be B group. The last one is small I. First parent is small I, even the second parent small I. The genotype is I small letters, both small letters. So the blood group, both it is recessive. So the blood group will be O blood group. Clear, children? So the next we are going to learn about test cross. So what is meant by test cross? It's a method devised by Mendel to determine the genotype of an organism. In this cross, the organism with unknown dominant genotype is crossed with a recessive parent. So here we are taking one unknown, we don't know, unknown dominant genotype is crossed with parent. How is the parent? Recessive parent. Clear? So in a monohybrid cross between violet flower, that is capital W, and white flower, that is small w, the F1 hybrid was capital W and small w. F1 hybrid, it was violet color flower. The test crosses are, you can see here two cases, case 1, case 2. In case 1, the parents you see capital W crossed small w, small letter, two small letters. Gamete formation, gametes are capital W, small w. F1 progeny is capital W, small w, that is violet color. In this case 2, here you can see what we are doing. We are taking this, but in a previous class I discussed. In a monohybrid cross, always after getting the F1 progeny, we will do self-pollination. But here what we did, we have taken this F1 progeny, that is capital W, small w, crossed with a recessive parent. Recessive parent, here the recessive parent is here. Clear? Okay. So here the parents, so one is heterozygous. You can see in the F1 progeny what we got, violet color, capital W, small w, that is crossed with recessive parent, small w, small letter, two w's. Then how the gamete will form gametes, you can see capital W, small w, the other side you can see small w. There is a crossing will take place, or multiplication, you can see capital W, small w. So that is heterozygous form, that is violet color. Okay, you can see here this one. Okay, this is heterozygous but violet color. Here you can see small letter WW, that is white color. So what you learn from this test cross, if all the F1 progeny are violet color, so in the first case all the F1 progeny are violet color, then dominant flower is homozygous. Okay, so here the dominant flower is homozygous. The second case, if the progenies are 1:1 ratio, here one violet, one white. Okay, so 1:1 ratio, then the dominant flower is heterozygous. Here you see this is heterozygous. Clear? So F1 progeny, or if it is violet color, then dominant flower it is. How is it? It will be homozygous. But if we are getting in 1:1 ratio, the dominant flower it is heterozygous. That is case 1, case 2. So that is called test cross. The next one, it is pleiotropy. So what is meant by pleiotropy? It's a phenomenon in which single gene exhibit multiple phenotypic expression. There is a one gene, but it's like abnormal gene. This gene exhibit multiple phenotypic expression. This pleiotropic gene affects the metabolic pathways, resulting in different phenotypes. For example, phenylketonuria, it's a disease I'll explain after this. Phenylketonuria is caused by mutation in the gene coding for the enzyme phenylalanine hydroxylase. The affected individuals show mental retardation as well as hair and skin pigmentation. So let us see what is meant by this phenylketonuria. Here you see, children, it's a phenylketonuria that we will study, and now I am explaining because it's related to that. It is one of the disorder or disease. It's an inborn error of a metabolism and is inherited as autosomal recessive trait. What is meant by autosomal recessive trait? It means it is a disorder or disease can be passed down through families. Clear? It's a type of disorder or disease. It can be passed down through families. 2 copies of an abnormal gene must be present in order for the disease or trait to develop. 2 copies of an abnormal genes must be present. So what is this disease? We'll see. I told it is an inborn error of metabolism and inherited as autosomal recessive trait. The affected individual lacks enzyme phenylalanine hydroxylase. So this individual is lacking one enzyme. Which enzyme? Phenylalanine hydroxylase. So what is the function of this enzyme? It converts amino acid phenylalanine into tyrosine in liver. So that is the function of this enzyme. It converts amino acid phenylalanine into tyrosine where in liver. So if this enzyme is absent, means what will happen? This phenylalanine it is accumulated and converted into phenylpyruvic acid and other derivatives. This affects the brain, resulting in mental disorder. Clear? So that is the disease phenylketonuria. The next topic I am going to explain here, that is polygenic inheritance. So what is meant by polygenic inheritance? Poly means many, genic means gene, many genes. So what it is? It's a type of inheritance in which traits are controlled by many genes or 3 or more genes. Such traits are called polygenic traits. So what is polygenic traits? It is a type of inheritance in which traits or character, it is controlled by 3 or more genes or many genes. That is known as polygenic traits. Here the phenotype reflects contribution of each allele and is also influenced by the environment. For example, human skin color. Suppose 3 genes A, B, C, capital letters, control skin color with A, B, C being the dominant alleles and small letters A, B, C being the recessive alleles. Then what will happen? You can see here the parents. Here A, B, C, always allele or genes we can see in the pairs. A, B, B, C, C, that is dark color. It crossed with small letters A, B, B, C, C, that is white color. From this, gametes forming, gametes A, B, C, other side small letters A, B, C. Then F1 generation will come. In F1 first filial generation or F1 generation, here you can see A, B, B, C, C, but it is heterozygous in nature. But the color will be intermediate color. So F1 generation, it is intermediate color. So one parent is dark, other parent is white, but the F1 generation is intermediate color. If you are going to F2 generation, we will have different types of skin tones or varied skin tones. Each type of allele in the genotype determining the darkness or lightness of the skin. Clear? So each type of allele or gene in the genotype. Genotype means internal expression, I told you. It determining the darkness or lightness of the skin. The next topic it is chromosomal theory of inheritance. This theory, it was proposed independently by 2 scientists, Sutton and Boveri. The full name it is Walter Sutton and Theodore Boveri in 1902. So which are the scientists? Walter Sutton and Theodore Boveri. In which year? 1902. According to this theory, since the sperm and egg cells provide the only bridge from one generation to other, all hereditary characters must be carried in them. So you know how the zygote will form, always you learned in the previous chapters. Sperm and egg, they are haploid in nature. They fuse together and will form diploid zygote. So they are the only one bridge from one generation to next. So all hereditary characters carried in them. Second point is, the hereditary factors are carried in the nucleus. So egg cell is single cell, even sperm another single cell. Inside the cell what is present? Nucleus is present. Nucleus, it also contains nucleolus and chromatin fibers. So this hereditary factors carried in the nucleus. That is the second point of chromosomal theory. Third point is, like the Mendelian alleles, chromosomes are also found in pairs. Here Mendelian genes or alleles, we told always we can see in the pairs AA, BB, CC, like that. So here the chromosomes also found in pairs. The next point is, the sperm and egg having haploid set of chromosomes. They are single cell. Okay, each one is haploid or single cell. They have haploid set of chromosomes. They fuse to re-establish the diploid set. So that is the next point in chromosomal theory. There are two more points. The next point is about the gene, where the genes are located in chromosome. Let us see. The genes are located on the chromosomes in a linear order. How it is arranged? Yes, there are 2 chromosomes of each kind in diploid cell. So how many chromosomes? There are 2 chromosomes of each kind in diploid cell. There must be 2 genes of each kind. In these 2 genes, each one in each of 2 homologous chromosomes. Clear? So there are 2 chromosomes of each kind. So there must be 2 genes of, it's like a pair, like 2 genes of same kind, and 1 in each of 2 homologous chromosomes. The last point is, these homologous chromosomes synapse during meiosis or meiotic division and get separated to pass into different cells. This forms the basis of segregation and independent assortment. So you learned in the previous class, third law of Mendelian laws of inheritance, independent assortment. Okay, so here this forms the basis for segregation and independent assortment. A gamete receives only 1 chromosomes of each type and thus has only 1 gene for a trait. The paired condition is resulted by fusion of gametes. The next topic it is sex determination mechanism. How can we determine or decide the sex of an organism? Okay, so here the finalization of sex, when it will take place, it will take place at the time of zygote formation. It is called sex determination. So what is meant by sex determination? Which decide the sex of an organism. It happens at the time of zygote formation. Okay, that time only we will finalize it. Finalization of sex, it will take place at the time of zygote formation. This is known as sex determination. This sex determination mechanism, there are 2 types. One is male heterogamety and second one is female heterogamety. What is meant by heterogamety? It means different sex chromosomes. You know, in the case of human beings, the sex chromosomes, that is, you learned already, in a somatic cell contain 23 pairs of chromosomes. It means 46. In that 46, 22 pairs are autosomes and 1 pair is sex chromosomes. Clear? So if you are taking male, you know there is 1 pair, it is XY, that is the sex chromosome. If you are taking female, you know XX, that is the sex chromosome. So here female heterogamety means different sex chromosomes. In female, you know XX, it means both are same. They are called homogamety. But heterogamety means different sex chromosomes. So which are the different types of heterogamety? In male we can see 2 types. In female also we can see 2 types. So in male, One is XXXY type, second one is XXXO type. In female it is ZZZW type, ZZZO type. Clear? So first we will see what is XXXY type. This type we can see in many insects and also in mammals including humans. Here males have one pair of sex chromosomes. That is, one is X, one is Y, along with the autosomes. So how many autosomes? There are 22 pairs autosomes and one pair of sex chromosomes. So total there are 23 pairs. It means 46 chromosomes. Clear? So here males have, so I told you this type we can see in insects and mammals including humans. So males have XY chromosomes along with autosomes, and females have a pair of X chromosome. Pair means XX. So if you are coming here, you can see the parents. Male AA, that is autosomes, plus XY. Female AA plus XX. So how the gamete will form? Gametes A plus X, A plus Y. But in the case of female, A plus X. Then third, F1 progeny. First filial progeny or F1 hybrid or F1 progeny will form. That is, when it cross multiply, you can see AA plus XX. That is female. The other side you can see AA plus XY. That is male. Okay. So this XX it is female, XY that is male. If you are coming to the second case, that is XXXO type, we can see in grasshopper, bugs, etc. Most of the time we can see in the males of some insects. Here males have only one X chromosome. Before we told males have one X and one Y. Here males have only one X chromosome. That is the reason we told XO type. Besides autosomes, and females have same like a pair of X chromosomes. Clear? So what is the one difference? Here males have only one X chromosome. Before it was one X and one Y. Here only one X chromosome. So we will write XO type. But the females one pair of X chromosomes. Then if you are seeing here, you can see the parents male and female. Male AA plus XO, because only one X chromosome, that is a sex chromosome. Other case, female AA plus XX. If you are form, the next gametes will form. The gamete you can see A plus X, A plus O. Other side you see A plus X. Then next cross multiply AA plus XX, AA plus XO. So this XX here, this XX, this is female, and XO, that is male. Clear? We can see in grasshopper, bugs, etc. this type of sex determination. Then next one is third one. That type it is ZZZW type. We can see in birds, fishes and fowls, etc. this type sex determination mechanism. Here what is the difference? Female have one Z and one W chromosome. So these are the sex chromosomes. One Z and one W chromosomes, whereas males have one pair of Z chromosomes. One pair Z chromosomes. Clear? So you can see here in the parents, male and female, male AA, these are the autosomes, plus ZZ. I told one pair Z chromosome. AA plus ZZ, female, AA plus ZW. Then gametes, A plus Z, other side gametes, A plus Z, A plus W. Then F1 progeny will form. Here AA plus ZZ. Then other side AA plus ZW. So ZZ, that is male, ZW, that is female. So this is third group. Then last group, it is ZOZZ type. This we can see in cockroaches. Okay, so here what is the difference? Here females have only one Z chromosome. Before we told there are two Z chromosome, but here females have only, here you see, one Z and one W. But here there is only one Z chromosome besides autosomes, and males have a pair of Z chromosome, that is same. So how is the parent? Male and female. Male AA autosome plus ZZ. Female AA plus ZO. Why they are writing O? Because only one chromosome, sex chromosome, only Z. Gametes A plus Z in the case of male. In female, A plus Z, A plus O. Then F1 progeny will come out. How is it? After cross multiplication, it will form AA plus ZZ. Other side, AA plus ZO. So this ZZ, that is male, because we told here one pair of Z chromosome, that is male. So AA plus ZZ male, AA plus ZO, that is female. So these are the four types of sex determination mechanism. So I'll repeat once more. First one is XX, XY type. Here I told female is XX, female is XX, male is XY. These are the sex chromosomes. Second type, XS, X0. Here female is XX, but the male only one X chromosome. So that is the reason we are writing XO. Then third type, excuse me. Third type, ZZ, ZW. Here females have one Z and W chromosomes, whereas males ZZ chromosome. The last type, ZO, ZZW, ZZ type. Here male one pair Z chromosome, and female there is only one Z chromosome. So we are writing ZO type. Okay, so the next topic I am going to explain about the linkage. This theory, linkage, it is discovered by T.H. Morgan. He carried out several dihybrid crosses in Drosophila to study the genes that are sex-linked. So let us see what is meant by linkage. Here you can see I drew a diagram. The people are standing together. Linkage or link means connection or connecting together between people or things or genes. Okay, here how the genes are connecting together, let us see. So here the physical association between genes located on the same chromosome, that is known as linkage. So what is linkage? Physical association between genes located on the same chromosome. So here you can see these are one pair of homologous chromosome. I named A. These are the genes, gene A and B. Here also you can see gene A and B. Here capital letters, small letters. So these are one pair of homologous chromosomes. These two genes, it's connecting or linked together. Okay, that is known as linkage. So there are two types of genes. One is linked genes. Second one is unlinked genes. So what is the difference between these two? Let us see. First one is linked genes. Linked genes means these genes present on the same chromosome. I told these linked genes present on the same chromosome. Okay, so even this A and B, you cannot tell these are not linked genes, or this A and this A, they are not linked genes. Here A and B are linked because they can link or connect together. Okay, that is called the linkage. So genes on the same chromosome A and B, these are called linked genes. Second point is they do not show independent assortment. So Mendelian experiment, we told the laws of independent assortment. We told all the gametes it will separate, they will mix in different way and forming recombinant or new combinations. Okay, so here A and B can link, but A and B cannot link. Clear? So that is called do not show independent assortment. It means the recombinations it will be very less. They both will combine and this will carry one generation to next generation, so on, like that it is going. Clear? Third point is they are inherited together. That I told just now, they are inherited together means these two going together from one generation to next generation. Clear? 2-3 generations is going together. Last point is they give a dihybrid ratio 3:1. Why it is showing dihybrid ratio 3:1? Let us see here. Here you think A and B are linked. Capital A and B are linked, you think. After that in F1 progeny or F1 generation we will get heterozygous. Capital A, capital B, small b. So which are the possible gametes? Here possible gametes are only two, capital A and B, because they are showing linkage. They cannot mix A and B or B and B. Like that they can't mix. So always possible gametes are only two, capital A, capital B, small a, small b. So these are only two possible gametes. So these gametes, it will go to the next F2 generation. Then we will get the dihybrid ratio, that is 3:1 ratio. But in the case of second one, unlinked genes, what is happening? Genes located on different chromosomes. Here, these genes located in the different chromosomes. And also another reason, sometimes the distance between the genes, it will be very far. So here they are very close. But in unlinked genes, the genes will be very far. The distance will be very far. At the same time, they are also present in different chromosomes. So that is the one reason for unlinked genes. Second one is they show independent assortment. Why they show independent assortment? Let us see. Here, in the case of unlinked genes, in the F1 progeny, we will get capital A, capital B, small b. Clear? So which are the possible gametes? Capital A, capital B, capital A, small b, small a, capital B, small a, small b. Clear? So there are 4 gametes. So these 4 gametes, in the case of independent assortment in Mendelian experiment, we told there is a, they are taken dihybrid cross means they are taken 2 characters, seed color and seed shape, round, yellow and green wrinkled. Okay, in F2 progeny we will be getting different types of recombinants or recombinations, because the first thing is it is dihybrid. It means 2 traits, 2 characters we are taking. The F1 it is heterozygous. Again gametes will form, same like 4 gametes will form both the sides and they will cross. So F2 we are getting many recombinants. In that 50% will be resembling the parents, parental combinations like parent, but the 50% will be new type. They are called new combinations or recombinations, or it is also called a recombinant. Clear? So these are the different 2 types of genes. Linked, I will repeat once more. So linkage means it is the physical association or connection between genes located on the same chromosome. I told you there are 2 types, linked genes and unlinked genes. What are linked genes? First, genes are present on the same chromosome. So this is one pair of homologous chromosome. Second, they do not show independent assortment because they are linked, so always they will go together in one generation to next. So I told in F1 progeny, this is a heterozygous format. From here, from this, these are the possible gametes, only 2 gametes. Third one is they are inherited together. They are going from one generation to next generation together. The next, they give a dihybrid ratio 3:1 because of here only 2 gametes. Unlinked genes, there are 2 reasons. One is genes located on different chromosomes. Second point is they show independent assortment. Here they do not show independent assortment, but here they show independent assortment because the possible gametes are 4. So they all will, in any way they can mix together, different way they will mix together. They are forming 50% like parent, 50 will be recombinants. So that is the next point. The next topic I am going to explain, recombination. What causes recombination, children? So the recombination takes place due to crossing over. So we should know what is, you already learned it. So what is crossing over? So I will explain to you how it is taking place. Clear? So crossing over means physical exchange of parts between non-sister chromatids of a pair of homologous chromosomes during meiosis. Okay, I will explain how it is taking place. So here you can see this is one chromosomes. You can see the genes are present in a chromosome. Here genes are the carriers of the hereditary or inheritance from one generation to next generation or from parent to children. Clear? So how it is taking place, this crossing over? I repeat, physical exchange of parts between non-sister chromatids. I will show you which are the non-sister chromatids of a pair of homologous chromosomes during meiosis. This crossing over occurs at a place. That place is called chiasmata region. That also you learned in the previous classes. Crossing over occurs at a chiasmata region. So I will explain to you. The first one, you can see this is one cell. Inside that you can see there is one pair of homologous chromosomes. Okay. So one red color, one is green color. So there is one pair of homologous chromosomes. In that you can see in a red and green there is a genes are arranged. So always the genes are same sequence of genes it is arranged. So these two red I marked, same like two green I marked, they are called sister chromosomes or sister chromatids. At the same time down I have marked one red and one green. They are called non-sister chromatids. Now you understood which are the sister chromatids. It means two red or two green, they are sister chromatids, and here down one red and one green, that is called non-sister chromatids. If you are coming to the second diagram, you can see here crossing over started. Always the chromosomes start to pair up. Okay, so one green and one red. Here you can see I circled also, it's a crossing over started. So that region, it looks like one X letter, that is called X-shaped region is called chiasmata. Clear? So that region is known as X-shaped region is known as chiasmata. So here crossing over will take place. What will happen during this time? Even genes also will exchange each other. Sometime some blending will take place even for the genes, and new recombinations will form. Okay. So here next you can see the third diagram, you can see now crossing over. You can see the green and red color is mixed. It means some parts of the green is attached to the red, some parts of the red is attached to the green. Same like even the genes also exchange. Even some blending of the genes also will take place. Clear? So what is happening? This is the reason for recombinations. If there is a more crossing over, more recombinants will form. Clear? So usually 50% like parent and 50% recombinants. But here there is a more crossing over, what will happen? More recombinants will form. Less crossing over, less recombinants will form. How it is formed? Suppose there is a more linked genes. Genes are very close to each other, you think. Genes are very close to each other and more linked genes, what will happen? Crossing over will be less. Suppose genes are very far, crossing over will be more. Clear? So if there is a more crossing over, more recombinants will form. Less crossing over, less recombinants will form. Now next we will see how the crossing over will take place between two linked genes. Okay, for that I want to show you some diagrams. First one is, you can see there is one pair of homologous chromosomes. Okay, one white color, one bluish color, that is one pair homologous chromosomes. In that genes are arranged. In one chromosome it is capital A, capital B. In another chromosome small letters A and B. Clear? Then during the time of meiosis, these chromosomes it replicate or duplicate and it will become two. So one chromosome it became two chromosomes here. You can see AAB, here also again one more AB. Same like this blue color AB, small letters again AB. So during the time of meiosis it will duplicate. Now you can see 2 and 2, 4 chromosomes here. They are called as sister chromatids. So what they are calling? They are known as sister chromatids. One pair of sister chromatids. Here also you can see one pair of sister chromatids. Again you come to the next diagram, third stage. Here you can see one sister chromatid in both the sides. It is not doing crossing over. But the middle you can see there is a one pair of sister chromatids started doing crossing over. It's coming in like X shape. That stage is known as chiasmata stage. Okay. So both the sides, one chromatid, sister chromatids, it's like same, but in the center there are two started crossing over. It is getting X shape, that is known as chiasmata stage. After crossing over, again the last diagram you can see. So I told both the sides, one sister chromatids, it looks same, but in the center two you can see, after crossing over you can see that chromatids, the parts exchange and the genes also exchange. Now you can see here capital A, small b, small a, capital B. Clear? So here now what happened? The parts of the chromatids exchange and also genes also exchange. So these are the reason for the recombination or recombinance. So how it will take place? We can take two cases. One is case 1. In case 1, what is happening? These are the first one I told, they are homologous chromosomes. In case 1, you think the genes are very closely linked. They are linked genes. So AB linked gene, again AB linked genes. Here the crossing over, it will not take place. They are completely exchanging into the next generation. Clear? So here no crossing over. So which are the gametes will form? Capital A and capital B, small letter A and small letter B. But in the case of unlinked genes or incomplete genes, what is happening? Here crossing over will take place in the second case. So first one is case 1 in the case of linked gene. Second case, that is unlinked gene or incomplete gene, incompletely crossing over will take place. It means you can see it will go to the first case, it will not go to the crossing over, there is no crossing over. But in the second case, it will go to the crossing over. It takes place. I told the parts of the chromatid, it has exchange, even the genes also exchange, and the new recombinations and the new recombinants will form. I want to conclude this topic linkage and recombination by saying two important points. One is 50% recombination occurrence. So what is that? Independent assortment of genes giving 50% recombinance is possible only under two cases. So which are the cases? One, first case is genes are present on different chromosomes. They are called unlinked genes. If both the genes are present on the same chromosome, linkage will take place and it will carry into the next generation. It's like inherited into the next generation. But in the case of unlinked genes, genes are present on the different chromosomes. In this case, 50% parental and 50% recombinants will form. Second case, genes are present on the same chromosomes but they are far apart. The genes should be very far. Okay, in that case, more crossing over will take place. If the genes are very far, crossing over will not take place and linkage will take place. Suppose they are very close, they are linked genes, the linkage will take place and they inherited to the next generation. The last point is the number of times crossing over occurred. It can be determined by counting the number of chiasmata. Chiasmata means nothing but I told that is a X-shaped part that the crossing over will take place. So the number of times, if you want to know how many crossing over take place, we have to count the number of chiasmata. Clear, children? So today I have completed codominance. The next topic I completed test cross, then pleiotropy, polygenic inheritance, chromosomal theory of inheritance, then linkage and recombination, sex determination mechanism. In the coming class, I will be teaching 3 more topics. One is mutation, second pedigree analysis, third one genetic disorder. Clear? So students, if you have any doubt, if you want to clarify something, you can mail your queries on dmcsonlinequeries@gmail.com. Thank you students.